Hello viewers, our today's topic is complementary genes. You know that each gene has its own individual effect. Even in a dihybrid cross, when we consider the inheritance of two genes together, their identity is maintained. They show their own effects. But sometimes the two genes they interact in a different way, and when both genes are present together, the two genes are present together, then some other phenotype is produced. So when the genes individually they have their own phenotypes, but when both are present together, then some other phenotype is produced. Then such type of genes are called complementary genes. Right? The example of the complementary gene comes from sweet pea. It is Lathyrus odoratus. In sweet pea, two types of flowers are there, two types of colors are there, purple and white. The flowers are purple when they produce a purple pigment, and that purple pigment is anthocyanin. The production of anthocyanin from precursor, it takes place in two steps. First, precursor changes into chromogen, then chromogen changes into anthocyanin, that is the purple pigment. For the conversion of precursor into chromogen, gene C is required. But for the conversion of chromogen into anthocyanin pigment, another gene is required, and that gene is P gene. So as far as the production of purple pigment is there, anthocyanin pigment formation is there for purple flower, both genes are required, C as well as P. So these C and P are complementary genes, because for the purple flower formation, anthocyanin pigment, both are required, so they are complementary to each other. Right? Now you just consider this particular cross. A white flowered variety is crossed with another white flowered variety. This is white because of the absence of capital P gene, and this is white because of the absence of capital C gene. But when these two are crossed in F1 generation, the genotype is capital C small c, capital P small p. In F1, C as well as P both are dominant, both dominant genes are present. So there is formation of purple flower because of the presence of C as well as P, both are dominant. When these F1 purple flower plants are self-pollinated, or F1 purple is crossed with another F1 purple, then in F2 generation this particular result is obtained. It is a typical dihybrid cross, as you have studied in the third law of Mendel, independent assortment. In the same way, the same inheritance is there, but phenotypes are changed. See, this particular line is of purple, this is purple, purple and purple. So wherever C and P both are dominant, then that will be purple, because for the formation of anthocyanin pigment, C and P both are required. But here it is white because P is recessive. It is white because P is recessive. C is dominant. C is dominant. Here also C is dominant. Capital means capital C dominant, but P is recessive. So again it is white. So 1, 2, 3. These three are white because of the absence of dominant P gene. These three are white because of the absence of capital C gene. And this particular is white because of the absence of C as well as P. Neither C dominant is present here nor P dominant is present here. So in this dihybrid cross, a modified phenotypic ratio is obtained in F2 generation, and that ratio is 9:7. 9 are purple. Which one? Which are having both dominant, C as well as P. But 7 are white. Out of these 7, 3 are white. This one, and this one, because of the absence of P dominant. And 3 other are white because of the absence of C dominant gene. And this particular is white because of the absence of both C as well as P dominant gene. So in this way, when two genes C and P, they have their same individual effect, but C and P, they have same individual effect, but when C and P both are present together, then they produce a new phenotype, and that new phenotype is purple flower. Okay, so in this way C and P are complementary genes. Okay, thank you very much.